We'll start our last talk of the session. We have a slight shift in speaker. Hua Wang will be giving the talk today, and we'll be hearing about identifying disease-sensitive and quantitative trait-relevant biomarkers, so biomarker identification, from heterogeneous imaging genetics data by sparse multimodal multitask learning. All right.
Thank you. My student, thank you. My student Hua Wang just got 2 babies 10 years ago, so he cannot attend this conference. So I will present this paper. In this paper, I will introduce our recent result on biomarker selection from integrated multimodal data. Sometimes we also call multidimensional data. So I think this is the state-of-the-art method to integrate different data sources. Recent advances in acquiring multimodal biomedical the neuroimaging and genome-wide array data bring new opportunities to study the influence of genetic variations on brain structures and functions. So research in this emerging field known as imaging genetics provide a greater promise of cellular biology of human brain to better understand the complex neurobiological system from the genetic determinants to cellular process and to the complex interplay of brain structure and function, behavior, and cognitions. So in recent research, several studies have successfully shown that the integration of genetic markers and neuroimaging markers can indicate the brain disease. For example, in 2007 papers, when UCI group combined ISMB study and brain structures, they find the highly correlated ISMB and brain structure to indicate the brain disease. Here, the correlation p-value of SMP to the brain structure is calculated, and the top p-value SMP is plotted in this peak. For those SMP now, it's very known they are correlated to different brain disease. The ADNI project, Alzheimer disease neuroimaging initiative project, was funded by NIH and several federal agencies and also corporations to collect different data set. In our study, basically, in this paper, we use the genotype, the genetic markers, and the phenotypes from the functional MRI, structural MRI, PET, and the DTI data. Of course, in ADNI data, we also have the protein markers and the DNA copy numbers. So based on those collected data, we are studying the cognitive behavior of the patient and also the disease diagnosis. For example, Alzheimer disease, MCI, and the health control. So that's the 2 different types of the genotype using in this study is the SMP. The phenotype we get from the brain imaging marker. The largest problem in analyze such heterogeneous data is how to integrate those totally different data sources. Actually, this is a very important topic in current bioinformatics research. For example, in TCGA study, we have so many data sets, how to combine them. To integrate those heterogeneous data is very difficult. This has been studied for many years in machine learning area. So far, the best method is using the multi-kernel method. However, that method actually is a little bit stupid because they assign one coefficient to one kernel. That means they consider all features from one data source will be treated equivalently. So that's why in research areas we don't see many people on using multi-kernel because even we simply concatenate all the features together, we can get a very similar result. So in this research, we designed a new model using the sparsity inducing norm to add regularization to combine different sources of data. So basically, we combine the ISMB genetic markers and also the marker from the structural MRI and also functional MRI and the PET. So we have two contributions in this paper. The first one, I will introduce our new method to integrate heterogeneous data sources. First, let's look at a very basic background for sparse inducing norm. So the sparse learning has been a very hard area in machine learning data mining area in computer science research. So the sparsity, basically here, because we are trying to select the markers, I don't talk about other sparse learning. So I don't talk about how to using sparse regularization to select biomarker, to select features. Usually we use the classification or regression model as a loss function. For example, using linear regression, logistic regression, or support vector machine. So Y is input data, the input of the data matrix. X is input of data matrix. Y is including the labels. Based on labels, we can decide the feature is important or not. W is a learned, is an output. W will tell us each feature is important, is significant to the study problem or not. So if we enforce W as L1 norm, so we will get a loss of sparse 0 in the W vector. Now we are talking about the simplest case, the two-class problem. So because let's consider if features with less weight, with small numbers, that means this feature is not important. So we shouldn't select that. Then if we enforce the sparsity, the L1 norm, then we can easily find the features with large weight. So these features are very important to the disease study, for example, classification or regression. So this model was proposed like many years ago. At the beginning, we have the L1 SVM. Later, we have the lasso, group lasso several method, where several model were proposed in statistics society. So if we don't use L1 norm as regularization, the standard like the ridge regression, least square SVM, we always using the least square regularization. If we use this regularization, the weight for every features, well, they won't have value. They won't have very. Excuse me. Most of them are non-zero values, so it's very. We have to rank them. Sometimes we may select the wrong features. So that's why sparsity is very useful in feature selection. However, now when we talk about multi-class, in last slide, I just show for two class. In multi-class feature selection, for each class we will have one feature, one parameter vector W. So typically we talk about parameter matrix W. So for each class we have one vector. So if we just enforce L1 norm like L1 SVM, so then this we typically call the flattened sparsity. So this is 0 point. The 0 value will go to everywhere. So this is not reasonable for feature selection. So typically we want to select the features that are important to all class or most classes. So based on this intuition, in 2007, the group, McJordan group in Berkeley, in UC Berkeley, they proposed structure sparsity regularization to select the features. They combine the L2 norm and L1 norm. So let's consider for one feature, if it's very important to all class, so it's not necessary to enforce L1 norm within this feature's weight. We should enforce L1, the L1 regularization between the features. So basically, they call the L21 norm. It's a mixed L1 and L2 norm regularization. So let's look at this figure to see. This is the W matrix, the parameter matrix. For example, for regression or for support vector machine, we always learn this W as a parameter matrix. this W will tell us each feature, each row is the weight for each features. For one features corresponding to different class, each column corresponds to one class. Here I use the cognitive score as a class. So each value tells us this feature is important to this class or not. So if we select a features, typically we want the features should be important to all class or most classes. So that's why we use the L2 norm for all the values within one row. Between rows, we emphasize L1 norm. Then for some features, if they are not important, then most of the whole row will become 0. So we can easily use this way to select features. So this is a very good method for feature selection. Typically, it's like a state-of-the-art method for biomarker selection. However, this method is still only for one data source. So now we are talking about how to integrate heterogeneous data sources. So the very naive way we can think about, for every data point, we just concatenate all the features together as a very long feature vector. Okay, if we properly normalize each data modality, the result will comparable to the multi-kernel learning method. So actually, it's not bad. However, if we just concatenate them together, then we don't use any interaction within the data, because some type of feature may be more important than others. We cannot treat them equivalently. So we should consider utilize interaction, the relations between the different types of features or within different types of features. So we propose a new operator function for multi-modal, or you can call it multi-dimensional data integration. So basically, our model built based on the loss function we can use using linear regression, logistic regression, or support vector machine. Here, I wrote the linear regression model, the simplest one. So the important one is the important point here is we have two regularization. The first one is the group 1 norm. Second one is 2-1 norm. 2-1 norm is similar, is the same as the previous slide. So let's see why we add these two regularization. Why after emphasize these two regularization, we can integrate the data sources. Let's look at this matrix. So this matrix is W matrix. Now for each data point, we concatenate all the features together. So that's like all the features here. So that's for class 1. So the first type of features, we have D1 dimension. Then we have second type of features. On TL2, we have K types of features. For each type of feature, we have several, maybe 100, maybe 1,000. So for every type of features, we emphasize the L2 norm within these type of features. Between different types of features, we use L1 norm. So totally, we have C class. So that's why we have C row here. So based on this group 1 norm, we can see if one type of feature is very important to certain outcome or certain disease status, so the whole class will get larger weight than the rest. Here I use the color bar, the color barcode to interpret the weight values. Each W here should be a 1 value. So if value is deeper, so the deeper blue will show us larger weight for the learned parameters. So that, for example, for this type of features, we can consider for the class C, they are very important. So this type of feature are less important for class 1. So based on this group 1 sparsity, we can learn which type of feature are important to which types of outcomes. And however, when we do a feature selection, we should consider even the whole data type, like all ISMB are not important, are less important, but still 1 or 2 ISMB are very important. So that's reasonable because the whole data set are not important, but still some of the markers are important. So we emphasize 2-1 norm in the other direction. So if one feature is important to most of the classes, then we still select this feature together. So these two regularization will learn which type of features are important to the final classification, then assign large weight to them. Meanwhile, we also consider individual feature. For individual feature, if the feature is important to all classes, we will also select them out. So for important class, of course, we will select more feature out. But for non-important class, we will select small number of feature out. So this two regularization will help us integrate different types of features and learn. After we get the result, we just rank them based on these parameter values. Rank them, we can select the features, the total features that are important biomarkers. Of course, this is a very general framework. If you have other data, for example, you have the graph data. Because we have the protein-protein interaction data. If you want to add the network information inside, we can add the graph. We can add the graph lasso as more regularizations. But it's for more specific study. So here we propose a very general framework. If you have other data integration, so you can add more regularization to incorporate domain knowledge. Although here we use the two parameter, gamma 1 and gamma 2 for two regularization. But indeed, in the real case, you can use one to replace both of them. Because you can consider both of them are important, are equally important. So you can use one, you can calculate their magnitude and using one gamma to combine them. So then you only need to tune one parameter, so it's more robust. So that's our new model to integrate multi-dimensional data. So now, in our study, in image genetic study, we have the second contribution in this study, in this work. We learned the heterogeneous tasks. This beside integrate different type of data, we also consider do the different disease and the disease and the cognitive behavior study. So in the regular case, for example, here we have the disease, the normal people and the disease, for example, Alzheimer disease people. In image genetic study, when people study the relation between gene and the disease, they first study the quantitative trait. The quantitative trait, for example, the cognitive score. Cognitive score is a score to tell people how the people memory change. Because this value is can tell us the dynamic change of human brain. So it's better than the final disease result. That's why usually people study the quantitative trait, for example, the cognitive score. First, find the related cognitive score to the disease and normal people. Then, based on this, the quantitative trait to find the genes related to them. Based on this study, we only select the gene 1, gene 2, gene 3. All three of them are important to the QT1 and 2. So we'll select these genes. However, if we only use the disease classification, only gene 1, gene 2 will encode the disease, but the Q3 only encodes the normal patient. So this Q3 should not be selected. So here we have two tasks. One task is the disease outcome classification problem. The other task is related to disease, but not still totally equivalent to disease. We have the cognitive behavior study. So here, this is two heterogeneous tasks. So in this work, we consider study these two problems together. We simultaneously study these two problems. We try to integrate the genetic markers and also phenotypical markers to find the most important biomarkers related to both cognitive behavior and disease. Because if markers are related to both of them, then these markers are more robust, the practical more robust, not just for statistically important. So that's why we combine both heterogeneous tasks to do that. So input, we still have all the image markers and the SNPs. The output will have two parts. The first part is the cognitive scores. Cognitive scores, they are continuous values, so we can use regression to do that. The disease outcome, here we only studied Alzheimer disease, MCR, and the normal control, so we can do the classification. So for the classification, we can use the logistic regression here. I skipped this part because it's very simple. We can divide the multi-class logistic regression loss function. And for memory score performance prediction, we use the linear regression, the simple linear regression. So because here we use the W as the logistic regression parameters, So we use the P as a replace W in the linear regression in case to avoid mix them. So Z is a label for in the logistic regression. So the simplest case, we can combine these two tasks. We do two tasks simultaneously. But when we do regularization, we will combine W and P and get a single parameter matrix, then to regularize this parameter matrix. So if we use the regular Frobenius norm regularization, so this one can be decoupled. It's not good. So we don't decouple them. We don't select a feature to be important to both tasks. So fortunately, after adding our multimodal data integration regularization, the group 1 norm and 2-1 norm, so the parameter cannot be decoupled. So that means we perform multi-class together, select features that are important to the multi-class. So that's our final linear model. So next slide, I will show our algorithm. For this model, actually, I published code in my website. If you look at the paper, I have the link. So our code we implemented to solve this problem. So because we do both classification and regression. If your problem is only regression or classification, you can just make either W or P as a zero matrix. Okay, then you can get rid of one task, just only do either classification or regression. So that's using our code, you can do your perform on your problem. We develop our algorithm using the related method. Here I don't go through the details. Okay, but we prove that people prove that this objective is monotomical decrease based on by our algorithm TL convergency. So the algorithm is very simple and it's quite efficient. So the implementation is very simple. If you look at our code, we use the MATLAB to write the code. It's very simple code. I skipped this proof for the conversion proof. You can find details in the papers. So we evaluate our data integration and multitask linear result using the image genetics data. We use the 4 image modality here with the 2 VBM and free surfer features from the functional, from the structural MRI, and also using the PET image features, and also we using the more than 1,000 SNPs. Totally, we have millions of SNPs. We use the Alzheimer genomic database to filter out this more than 1,000 SNPs. They are highly potentially, excuse me, they are probably correlated to the brain disease. Some of them will already be proved they are very important, but most of them they didn't prove. That's why we still need to do the biomarker selection, find which one is the most important. For memory score, we test on 5 different on 5 different memory score systems. Basically, they always ask people to read different numbers or letters. Then after a certain time, they ask them to come back to test they can remember or not, give the score for them. So the disease status, we use the health control MCI and Alzheimer's disease. That's a number of subjects that we use. So here we show the biomarkers for the 4 different types of features. For example, like in free surf data, most of them we get almost 0. Only some of the important one we we selected they get a large weight. And actually, these features are very important. They have been proved to be important in the literature. For SMP, we didn't list all of them. We listed the top 15 SMP we selected. For example, the first ApoE has been proved. This one is very important in brain disease. And also the BIN1 and the PICM also have been proved in these 2 years new publications. So the rest of SMP, we are verifying them in our ongoing work. So we map the neural imaging markers on the brain regions to see which regions are related to which region provide the important biomarkers related to the Alzheimer's disease. So we compare since we did both heterogeneous task classification and also regression, so we compare to the state-of-art method to see how much improvement we can get using our method. So for disease classification, for Alzheimer's disease detection, we mainly compare to the baseline SVM method, and also a lot of multi-clonal learning method, because the multi-clonal learning has been studied for many years. So we compare our method to this multi-clonal learning method. We can see we have very large improvement, get a very good result. So, and also our method is quite robust. We don't tune parameter very much. We just, in very large range, we get a very stable result. Meanwhile, we also compare the memory score prediction result with the standard multivariate regression and the region regression. After we integrate all the features, we get these values are the distance, the memory score distance. So the smaller is better. So we also get a better result in memory score prediction. This work has been done between the UT Arlington team and the Indiana School of Medicine team. My student Hua Wang and Fiping Nie, and also our collaborators from the IUPI team, we work together, design the algorithm and perform experiments. We send to all the funding agencies to support our research work. I'd like to thank to our audience. This is the last talk today. I'm very happy we have many people here. Yeah, please go ahead, download my code. If you have multiple data for integration power, yeah, you can test them. Thank you.
Great. Well, let's thank our speakers. And we have time for 3 questions before we go upstairs for the final keynote speaker of the day.
Yeah, thank you for nice work. I'd like to have you take a look gamma 1 and gamma 2 regularization parameter after you running it, and what's the value of gamma 1 and gamma 2?
The gamma 1, gamma 2, typically if you set up from 1 to 100 should be okay.
Pardon?
From 1 to 100.
Oh, you said 1 to 100.
Yeah.
Wow.
Yeah, it's okay.
Okay.
It depends on the value of your features.
Right.
So if you normalize them from 0 to 1, so then you just set up around 10 should be okay.
Right. So you tune that parameter through the cross-validation, right?
Yeah, yes,
right.
The cross-validation.
Right. Okay, thank you.
Hi, it's very impressive work. So I'm just wondering, have you applied your model to, for example, the gene expression data? Where in that data set, you have very high dimensionality, right? You have like 20,000 features, where the sample size is very small. So usually you have a kind of overfitting concern. So have you actually, experiment with that?
We didn't try that because this one also we have. That's why we use the feature selection, because we want to avoid overfitting. So that's why we get the feature selection model. Like here, we also have very large dimensions. So of course, you can apply to the gene expression data. Because ISMB also has very large dimensions.
Right.
Yeah, we didn't try, but you can test them. I think this should be better, much better than multi-clonal learning method.
Okay. So have you compared your model with the neural network? It seemed like to me that neural network might actually have more advantages over SVM or some other type of model.
Yeah, actually, neural network most likely will overfit the model.
Okay, right, maybe, okay.
That's why usually I don't use the neural network. They always overfit the model.